Good morning, good afternoon, everybody. So I'm Jérémy Saunier, Catalan Vice President and Chief Scientific Officer. I'm currently leading Catalan scientific community and technology innovation effort across our small molecule, biologics, cell and gene therapy field. Today, I will share with you what we believe is the expanded role of a CDMO in the field of cell and gene therapy. Obviously, you should take any forward-looking statement in this presentation in light of this disclaimer. So let me start by saying that at Catalan, we really love the dynamic of the cell and gene therapy space and the disruptive innovations that are on the horizon. So Catalan first entered the gene therapy space in 2019 through the acquisition of Paragon. What we really love about Paragon was their deep expertise and capabilities in viral vector manufacture and specifically AAVs. Earlier this year, we added cell therapies capability through the acquisition of MasterCell, with the ambition to provide a hand-to-hand integrated solution to the cell and gene therapy customers. The MasterCell team brings amazing experience and capability across a very large range of cell modality that are not just CAR-T. They have experience with the most complex cell modalities such as iNK, iPSC, and also implantable cells and more. So the thing that is exciting about cell and gene therapy for Catalan is the promise to cure patients and not just to help them. And as Catalan has a very high focus on patients through our patient-first culture, by stepping in the space, we feel like we took a responsibility there. Also, personally, there is no better time and no better place than Catalan to be leading our science and innovation organization in a world where molecules, clinical trials, and modality are growing more complex. Each of 2,000 scientists is very well aware of their mission, enabling the emergence of new therapeutic modality by providing access to scale and to manufacturing technologies. So today, I'm delighted to share with you the mindset, the approach, the structures, and the technology we put in motion to partner with Integrators on their journey from discovery to first proof of principle in humans. So when we take a look at the horizon of new therapeutic modality emerging in preclinical or clinical development, there is a lot to be optimistic about. It is easy to be excited when we see this modality opening up approach that could not be tackled by small molecule and monoclonal antibody. Indeed, only a very small portion of the protein target are currently attainable with small molecule or mAbs. And these new emerging approach open a wide range of new mechanisms, activating new cellular function, replacing defective genes, and affecting the whole system rather than a target. With cell and gene therapies having now reached patients and obtained approval, we have several reference points that lead us to believe their adoption pattern will follow the one we've seen with mAbs and potentially at an even faster pace. Indeed, once the first modality reaches the patient for the first time, many of the barriers go away. Faster than ever, our scientific community is able to adapt and learn. Therefore, companies like Catalan, ready to adopt and incorporate new modality, will likely do really well. So the speed at which cell modality are evolving, specifically in complexity and specificity, is amazing to see. So we're going to look a bit deeper at cell therapies, and specifically the next generation autologous CAR-T treatments are all in an advanced stage of clinical developments, either targeting over antigens or favoring over phenotype or integrating kill switch mechanisms. So in the footstep of those autologous therapy, we see also gene editing technology that have propelled several allogeneic programs to the clinic with multiple edits to affect graft response, graft host rejects, or exhaustions with PD-1, or even targeting solid tumor with CD70 and others. Beyond that, the therapeutic approach continues to diversify, working on a broad range of cell modalities, including NK cell, tumor-infiltrating lymphocytes, macrophage, iPSC, and also implantable encapsulating cells that are acting in vivo as protein factories. We also see companies like Rebush emerging, leveraging red blood cells to impact our immune system, either activating it to fight tumors or retraining it to tolerate self-antigens. Whatever the modality they are pursuing, every innovator faces a common need that is often overlooked. How do they ensure those innovations are developable, scalable, and ultimately manufacturable? So for this new modality, more than traditional therapy, the process is often and often remains the product. So over this presentation, we will cover a broad range of recommendations to ensure the manufacturing technology works in the clinic and works at a commercial scale. These recommendations will be evidenced by selected examples. First, how to partner earlier with innovator and conduct sound developability and manufacturability assessment to get started on the right track. Secondly, we will focus on the importance of manufacturing infrastructure and environments, which have to be flexible and multi-purpose to custom design a reliable process. Third, we will remind the must-have integrated toolkit of technologies as a license to operate in the space of cell and gene therapy. And fourth, we will propose a methodology to translate research-based manufacturing processes into clinical and commercial worlds. So let's speak first about developability and manufacturability assessments. According to Deloitte's analysis, the return on investment from small cap pharma overachieved over the past 5 years, return on investment from the 12 large cap pharma. However, these overall numbers hide some key imprecisions when coming to the early days of developing new therapies. Over the past years, we've seen at Catalan that 3 out of 10 candidates from small cap innovator, either in the small molecule, biologics, or cell and gene therapy segments were by design not developable or manufacturable, and several of those approaches needed to be re-engineered with a minimum 6 to 9 month loss on average. So therefore, we ask ourselves the following question: how could we partner earlier with innovator to help them design their development and manufacturing strategy and avoid all the pitfalls overlap encountered when bringing new therapies to scale? So this requires a change in the way we partner. We've set a specific group of experts dedicated to work with innovator on an open access model. These experts' mission is to freely interact early and often upon company inception to advise innovators on the pathway to develop reliable manufacturing technologies for their product. So let's remind that in several of those cases and instances, the starting point could be as thin as a process developed in research and academic setting. For new and more complex modalities, our experts lead a comprehensive diagnostic process which emphasizes with emphasis on several elements such as materials, processing steps and controls, environmental handling, quality controls, and many others. So now let's exemplify this in the case of cell therapies. Most cell therapies are currently undergoing early phase and academic trials with manual operation, extensive bill of testings. And this reinforces the need to conduct those open access diagnostics earlier and characterize the distance that separate innovator from clinical trials. We're covering here an example of a diagnostic conducted some years back in time from an autologous TCR cell therapy, which we are going to follow up across the presentation. So through the diagnostic, we identify several areas of evolution. We've been able to select the most optimal known equipments to reduce manual operations, variability, and open step. Listed the 18 CPP which could be uncontrolled or varying, and find reliable alternative to several ancillary material across each processing steps. So let's take a few examples of recommendations we issue for this specific TCR process. So starting with leukapheresis product treatment. The starting isolation and purifications methods, fecal and immunomagnetic separations there, are quite straightforward in labs, right? However, they cannot apply similarly in CGMP environments as they present large variability between operator, a large number of open steps, and are only applicable to small volumes. So in this case, we've evidence that isolation with CPACs could be skipped to run straight into a prodigy selection process and keep processing time well below 12 hours, as was the target to preserve the cell. So moving to the activation, transduction, and expansion steps, this is where we had the most varying and uncontrolled CPPs, right? For these steps, we recommended to replace polybrand and centrifugation that are used to concentrate the vector and cell, which is less than ideal for CGMP, and replace it by a retronectin-based process. Looking now at the process parameter for this product, it was quite key to define the optimal condition to get the right TCR expression in competition at the surface with endogenous TCR, and at the same time control viral copy number to limit risk of altered insertion that could trigger oncogenes and many others. So therefore, we fixed most parameter, including cell concentration, and recommended to link in a DOE retronectin with MOI and cytokines. And we could even check if we could limit IL-2 to preserve the memory potential of these therapies. So this enabled to highlight in a 4 weeks of work period an expected 14-month pathway to reach a clinical ready stage. It could have been painful to make all those changes post-clinical due to a complex CMC bridge to demonstrate equivalency between processes and product. So as shown in the diagnostic, for a brand new product, which could be as specific and diverse as red blood cell or iPSC, we are able to engage in early discussion with innovator to propose optimal non-equipment to be tested in development setting. We recommend revised phasing of steps to reduce manual and open operation, and we isolate key parameters to generate process understanding, and we revise ancillary material that have been better changed for cGMP manufacture. This slide answers the question, how do we enable those next process understanding study? And as a second enabler, we believe that executing upon those plans to deliver a reliable and scalable process require a flexible and adaptable manufacturing environment that enables to put all pieces together to develop the modality. We have been in the past leveraging regularly flexible manufacturing suite to support our partner in enabling modality to reach first clinical milestones. We did so with Moderna for the first messenger RNA cancer vaccine reaching the clinics, and CRISPR Therapeutics and their innovative gene editing allogeneic platform. The concept obviously includes separated suites for all recombinant viral vector manufacture, separated cell therapy suites, and more generic suite for any recombinant protein, biomaterial, or for specific delivery systems such as fill-finish devices or devices, nanoparticle-derived technology, or even encapsulation of cell technology. And those systems will be used in further processing of some of the modalities. So when developing those processes, often equipments and manufacturing platforms are being repurposed and customized from pre-existing processes. So therefore, equipment are the movable variable in your model. More easily handled to move those in development settings, but also we know the way to handle those in grade B settings for the cGMP runs. So to execute all those plans, innovators need to access as a third enabler the right must-have toolkit of development, delivery, and manufacturing technology. And they need to continuously invest in R&D to innovate and evolve this toolkit. So for the gene therapy field, through the acquisition of Paragon, we now have leading viral vector expertise and capabilities. From a technology standpoint, 80% of all projects are now being handled using a triple transfection process with adherent or suspension to 93 cells. To further those programs being handled as a suspension process where we early adopter of the trend we see in the manufacturing technologies, and overall bringing us success, bringing it to scale for over 60 programs. Also, in a desire to simplify the manufacture, we are leveraging bacterial manufacture for the CRISPR-derived therapy, which often get associated with AAV for gene of interest delivery, so endonuclease. For the cell therapies field, MasterCell joining Catalan earlier this year brings unique expertise and assets across a broad range of cell therapy modalities, and we really love the experience in toolkit of manufacturing technology that MasterCell team have developed over the years, which range from, as we say, CAR-T, TCR, iPSCs, and more complex systems such as implantable cells to be encapsulated before. So we're currently integrating this extremely talented scientific team to form of MasterCell and Paragon to form an industry-leading end-to-end offering in the cell and gene therapy space. So meanwhile, our R&D teams are acutely aware of the importance to innovate and develop distinct capability in emerging space to simplify and accelerate paths to clinics, such as new iPSC-derived technology we're developing, but also they remain open to partner on disruptive technology areas, such as stable producer cell line for AAV and for viral vector more generally, and fully closed therapy cell therapy platform. And we know those will completely change the economic of those fields and benefit the whole space. So all of this goes with stating the importance of bioanalytical platforms, which have been evolving in complexity and specificity, gradually moving from recombinant protein, gene therapy, and then cell therapy. Now, we have a proper diagnostic plan in place. We have a flexible and multi-purpose environment that our needs, and we have access to the right toolkit of technology. Let's put this in motion and move to the development and the CMC world. By nature, these therapies are transforming the way we approach diseases. also the accelerated time to regulatory approval require us to think how we develop those treatments and cure, and even rethink those. In Kemria case, the first patient had been enrolled in phase 2 study August 2015, and 6 months after, with 34 enrolled patients, response data for 23 patients, and overall 82% remission rate, breakthrough therapy designation was granted. Then everything accelerated, and 18 months after first patient enrollment, BLA was submitted, and 6 months after, approved. And this set the standard as 2 years from patient enrollment to market launch. If we put it in CMC words, 24 months to move from a phase 2 ready process to process understanding, process characterization, scale up or scale out, and PPQ or CPV, like in the event of autologous therapy, is a very short time. In Zolgensma case, the timeline were a bit more advantageous, but nowhere close to traditional therapy. Phase 3 CMC plan started to be discussed in a Type B meeting May 2017, 18 months after BLA was submitted and 6 months after approved, based upon 15 phase 1 patients and readout from 21 patients enrolled in the phase 3. So again, 24 months from phase 2 ready process to scale up and PPQ readiness. So in this abbreviated time, how do we ensure meeting product quality as well as process efficiency targets? At Catalan, we always link a quality by design concept to a manufacturing by design concept, where both critical quality attributes and critical manufacturing attributes are considered equal in process characterization studies. Manufacturing by design takes the angle of optimizing process efficiencies, economics, and supply chain management. Or manufacturing by design study coming along with quality by design study for allogenic cell therapy points in the direction of fill finish, cell expansion, and purification of raw material strategy for improvement. So now to illustrate this, I would like to come back to the TCR example that we discussed earlier in the diagnostic phase, and want to detail how these plans translated into real process understanding studies. So the aim of the DOE that we executed and we're going to present now was to define conditions that enable us to achieve the best TCR expression, which is in competition with endogenous TCR at the surface of the cell, with controlling at the same time viral copy number, typically keeping it below 5 TCR class 1 and maintaining cell viability. Sometimes some vectors are shown to be suboptimal to express surface TCR and need to achieve high multiplicity of infection, which could have an effect on viral copy number. So here we study the effect of cytokines, multiplicity of infection, and the change to the retronectin-based process, as you might remember, that we recommended from the diagnostic to move away from the polybrand-based process. And we identify a positive space with retronectin and keeping the multiplicity of infections above 3. So this second TCR-derived dataset aim here to evaluate the effect of starting materials, either fresh or frozen cells, and checking if removing IL-2 will be possible to theoretically preserve memory potential. So it's a key point of focus for many. So in this case, data showed that only fresh sample could enable us to have a proper space to operate. But there is a very narrow spot only for IL-2 removal. So this is often true, but this exemplifies the typology of the data we could get through some process understanding study as early as possible in the development cycle and preferentially before jumping to the clinics. So in all of these studies, manufacturing by design parameters have also been incorporated. In conclusion, we really love the dynamic of the cell and gene therapy space, the disruptive innovation on the horizon, and the promise it carries for patients. At Catalan, we understand this space is rapidly evolving, and we foster a change-by-minute philosophy to continuously evolve our technologies, partnering models, infrastructures, and expertise in the cell and gene therapy field to lead through those change and fulfill our ambition, powering the innovation and growth of the life science industry by becoming its leading development and commercial partner for advanced technologies, first to scale innovation, integrated solutions, and new therapeutic modalities. So this is why our dedicated open innovations teams are days and nights and always on the watch for any new technology innovation partnership or collaboration proposal that you might have in the field of development, delivery, and manufacturing technology. Therefore, we invite you to submit any proposals at catalan.com. Again, thank you for your attention.